So we're doing interviews on mycological history, and we have Tom Harrington here. I'm Don Pfister.
I'm Meredith Blackwell.
I'm Tom.
And Tom is at the University of Iowa.
Iowa State University.
Okay.
Yeah.
Yes, I looked that up the other day, and it's very confusing because the official name of both schools has state in it. Online.
Our sister school is University of Iowa, right? Is that what you
But
mean?
it's also got, well, we'll skip it. I'll tell you later.
Okay.
So one of the things that Meredith has established on doing these interviews is to talk about your early life and kind of how you got into mycology.
Oh, well, I mean, it's kind of a long story. Should I do a long story?
Well, start
Sure.
early. Wait,
Okay.
you may be starting too
Well, how
late.
about when I got out of high school?
When you were born?
No, well.
What was your family like? Did they encourage you
Oh.
to be a mycologist? That kind of thing.
not-
Early.
It really wasn't relevant to
Okay.
mycology, but
Okay.
so I grew up in Omaha, Nebraska. My folks are from Western Iowa, and so after World War II, most of the people in Iowa moved to the nearest city, and so that's where I grew up with a bunch of ex-Iowans, and we weren't a very academic group. I was the oldest of 5 kids. I went to a Jesuit high school, which was
I didn't
pretty
know that.
costly for a lower middle income family, but so I learned a lot there. But when I got out of high school, I wasn't really ready for college yet. So I went for a couple
The
semesters.
Jesuits
and
didn't
tried-
get you ready?
You know, they did academically, but you know, it was a unique time then.
Okay.
So I graduated in 1970. So that was the era of sex, drugs, and rock and roll, which was more important than
All three.
university life, you know. So, but then I met Susan, and we got married when I was 20 years old. So I missed the whole sexual revolution thing. The drugs I really shouldn't go into, but the rock and roll was really cool.
Were you a
And-
musician?
What?
You're not a musician.
No,
Okay,
no, I couldn't
but-
play. I just liked it. So we went to a lot of concerts and stuff like that. And but we decided Omaha in the Midwest was too boring, so we went off to Colorado, like many people did from Iowa and Nebraska when they grew up. And I wanted to figure out a way to live and work in the mountains instead of Denver. And so that's when I decided to go back to school. I had a neighbor woman who taught at community college at Denver at the time, a young woman, and she said, you know, you need to go to Colorado State, get a degree in forest biology, and then with that you can get a job with the Forest Service. But go to community college at Denver, get all the cheap credits you could, and Colorado State had
Move 'em
an
over.
agreement with the community colleges that they would transfer all credits.
Sure, yeah.
So that's what I did. So I went to community college at Denver for a couple of years, got a lot of credits, and transferred up. In the meantime, they changed the forest biology requirements for a degree, but they went from a quarter to a semester system. So it was going to take me almost 2 years longer to graduate than what I thought. So I went through the catalog and I saw, oh, if I got a degree in plant pathology, I could
Get out.
graduate in 3 semesters in a summer. So I didn't really know what it was, but that was my major. So that was the motivation, because once I got into plant pathology, I took a course in mycology that I like.
Who taught it?
Brent.
Brent. He was treasurer of
Yes.
Yes, that's who. I can't
Brent.
remember his last name, but
Oh,
Brent was pretty enthusiastic
Yeah.
teacher, and I liked that. And then I decided to go on for a master's. I needed that to get a job, and forest pathology was really what I wanted to do. And then I worked for the Rocky Mountain Research Station and for the Forest Service at summer in between. And Frank Hawksworth was my main boss.
Oh, the
but he's a-
mistletoe guy.
Yeah, dwarf mistletoe guy.
Oh, I've
and
met
he was a
him.
terrific guy.
Oh,
So
yeah.
he's kind of my first mentor. And then I worked for Tommy Hines, who was also a forest pathologist there, and he had a project on aspen cankers. And so that was a pretty cool job too, surveying diseases in aspen stands, beautiful forest. And so he introduced me to Stratus cystis, which kind of became a lifetime interest that I had. It was quite a few years later when I got to work with Stratus cystis when I really got into it. I mean, it was a cool fungus. They inoculated these trees and big ugly cankers developed, and the fungus had parathecia that would form and culture readily and have a sticky ascospore drops
with
for the
long
insect.
necks and
Yeah,
so
the
forth,
long neck.
Insect dispersal.
Yeah, and then a fruity aroma, kind of banana-like, that would attract insects. And so I thought that was pretty cool. So this kind of jumps ahead, but about 25 years later, I had some NSF funding to look at Stratus cystis, and I had a student that looked at North American species, including that one that causes aspen canker. So it was known as Stratus cystis fimbriata, but the student, master student, Jason Johnson, showed that it was a really distinct species, and so we named it Stratus cystis populicula because it was on poplars. And I guess we weren't careful enough because somebody had used that name for an Ophiostoma fungus many decades earlier, and so the name was occupied and we had to come up with a new name.
After everything got combined.
After we published it, then we realized, wow, we made a mistake. So we were working, I was another student from Brazil, and we were working with a related fungus, so we had a manuscript and we were going to add in a new species name. So you have to give it a whole new name, right?
Yeah.
And so we were going to call it something else, and then there was a rival group down in South Africa, Mike Wingfield's group, and they were working with these fungi too, and they had another Stratus cystis paper where they just mentioned, they just gave it a new name, you know? So they didn't have to do any work. They just had to refer to our earlier typification and stuff, and all they have to do is add in like 2 lines, and that's their new name. So I'm sure they knew I wouldn't like that, you know, that they were scooping us on renaming it, but them's the rules, you know. So
we were talking about Lloyd.
Yeah.
Lloyd yesterday, C.G. Lloyd, and maybe that's what got him up on this tear. Maybe someone did back to him.
He called them the advertising pages, where people had named those names, and then they get their name, you know, credit for the species or combination afterwards.
Well, I think you probably got enough
So they
names.
beat you to it.
Yeah. So but guess what they called it?
They name it for you?
Yeah, Stratus cystis harringtonii. So they thought that would soothe me. I was still ticked off. But it's interesting, that was the first fungus I really got fascinated with. And then in the end, because of my mistake, it ends up being named after me.
That's good.
So it's a funny field, isn't it?
Yeah.
Okay, so let's go back then. So after you left Colorado.
So I finished at Colorado State, worked that summer for the Forest Service, and then I went to Washington State University. They had a good plant pathology department there, and they had 3 mycologists on the staff there. So there was Gardner Shaw, Jack Rogers, and Reuben Duran. And
you worked with Shaw.
So Shaw was supposed to be my major professor.
Oh, okay.
And Gardner got a message back to me to Fort Collins, because I didn't have a phone and it wasn't that easy to get a hold of people. You know, you had to write letters, and this was urgent. So I had to call him up, went to the phone booth and a bunch of nickels and called him up, and he goes, really glad you're coming up. It'll be great to have you here, but I'm going to be gone for 2 years. While you get your master's degree, I'm going to be in Amman, Jordan. And so, and I thought, you know, that doesn't sound too good. But he goes, you know, I have a grad student here who is going to finish up in a year, but he's going to stay longer, and he'll take care of you as you get through. And I thought that was pretty dicey, but it was the best thing that ever happened to my career. So that was Bob Blanchette, was the student that was going to hang around. So he was a PhD student, I was a master's student, and they let us do whatever we want. And Gardner, the professor, was gone, you know, so they were tolerant.
How
and,
much fun.
Yeah. So I had 3 courses, graduate level courses in mycology. I took a fungal genetics course from Ed Tulucky over at University of Idaho.
He was an Alexandros student.
Oh,
I used to
Ed
see his
was a
picture on
great
the wall.
guy. So we just went through Fincham and Day's genetics book from
that
the
was-
front to the end.
That was the one we used.
And it was all, the era of using different phenotypes for genetics. There was no DNA work, hardly any protein analysis. It was just strict phenotypes. So, but I got into that. I thought it was pretty cool. I worked on a wood decay fungus, Cryptoporus vulgaris, that was associated with bark beetle attacks. And I used like dikaryon, monokaryon pairings to identify different wood decay fungi and stuff.
What did you find out with Cryptoporus about spore dispersal?
Well, so this is a polypore fungus, and it fruits where the bark beetles go in and out. It goes, so it gets into those tunnels and comes out. And when it comes out, there's a covering below the pore layer that holds back the spores. Eventually, there's a little hole that develops down below. And insects will come into that cavity where all the spores are, and so people thought they were picking up the spores and taking them somewhere, but they had to take them to a bark beetle gallery. And of course, the bark beetles wouldn't go to those fruiting bodies. So it was pretty unclear how it was being dispersed. And there were some other ideas about how it was dispersed, and then I think it was Jack Rogers who suggested, he goes, this could be wind dispersed, how do you know? And so we put funnels underneath the fruiting bodies and collected all the basidius spores that dropped through that natural covering, and most of the spores escaped.
Yeah.
And so the fungus is really prominent where you have lots of bark beetles on pine or Douglas fir or some other host, and also areas where it's dry during the summer.
I remember
so kind of a Mediterranean type climate.
Found
is
a
my favorite.
ton of it at Berkeley off the campus when we were there, and that's the first time I'd ever seen it.
So it's across the U.S., but most common in the West where there'd be dry summers and cool, wet winters. And so I guess we hypothesized that covering below the pore layer was to maintain high humidity and stuff, so during dry summer months. And that's when the bark beetles are active. And so it was a good way to produce basidius spores. When the bark
Well, the
beetles
reason
are
I
out.
wanted to bring this up and to have you tell how these are dispersed, that they probably are air dispersed, is
I
that
think
just
so.
the other day someone asked me about this, and they had
Oh,
the
yeah.
old idea about the insect dispersal.
Yeah,
So I referred them to your thesis work.
Well, that's a pretty
was-
obscure piece of work.
Well, it was
but-
in the last 2 weeks, actually.
Yeah.
Someone was concerned about it.
Yeah, so I think that was probably my first paper.
So you did that. That was part of your master's.
Yeah.
And then what?
Well, then I wanted to get into more forest pathology that was oriented with killing trees and stuff. So I thought, killing trees was pretty cool. And I did pretty well on my master's and stuff. And, I could do this. I could go on for a PhD. And it was really Bob Blanchette who really encouraged me to move on. And so at that time, at least in the western U.S., Berkeley had the best forest pathology program with Fields Cobb and Dick Parmeter, and so that's where I went for my PhD. Fields Cobb was actually my major professor, but Fields and Dick had a shared lab, a pretty big lab, kind of a complex of labs, and all the students and technicians and stuff were
Kind of
pretty
together.
tight group.
Yeah.
Yeah. So I worked on black stain root disease, leptographia, Wagneri, and there were 3 different kind of types of this fungus. They were pretty subtly different in phenotype, but we didn't really have good phylogenetic analyses then. So we described 3 different. There were 2 new varieties and an old one that were on
Okay.
different hosts.
Well, let me ask you, did they have different aromas?
No, so these leptographiums are in the Ophiostoma group
Right.
that aren't aromatic.
But the reason I'm asking you is, well, go ahead and finish, and I'll tell you why I'm asking.
Well, go ahead and ask me.
Okay, there's some people at Auburn who are training dogs,
Oh, yeah.
and they're training dogs to do all sorts of things, find reticulated pythons in the Everglades so that people can get rid of them and that kind of thing. And they claim that these dogs could also sense, one dog in particular, 4 different leptographiums. I think it's 4 from above ground. And I just wondered if you thought that was possible. We don't know what the dog.
You know, I don't know. They can smell fungi apparently. So I know. So we have a project out in Hawaii on a ceratocystis that's killing the native ohia trees there, and they brought these dogs out from Florida to smell these trees.
They
And
made
so they
them
go
hard.
around and they're sniffing trees, and they don't know if they're diseased yet or not. So we'll see what the data looks like.
Yeah.
So I'm a little dubious.
Yeah.
You know, when a tree is infected with a real pathogen, like these leptographium in the southeast aren't really aggressive.
Yeah.
There's a question whether they really cause disease or not. So there's like root-feeding insects that bring these fungi in, and so the fungi may be incidental. And they're not nearly as aggressive as leptographium wagneri that causes the black stain root disease. If you have an aggressive pathogen in the root system, the host volatiles are just going to be through the roof.
Yeah.
You
Yeah,
know?
maybe that's what they're
Maybe,
sensing.
Yeah.
But
So
whether they're differentiated.
Yeah,
would
they
be
had
the
different
question.
behaviors for each species, so they
Yeah.
acted out.
Well, that's pretty cool.
Acted out their
I haven't seen a
answers.
lot of hardcore data on discerning subtle differences in species that are like closely related or not. But yeah, it's an interesting area, I guess.
Yeah. Do you have any pictures of the dogs? I
No.
want one.
No.
Oh,
I-
okay.
So Berkeley,
Yeah.
PhD.
Okay, so I about got my PhD, and you know, there was, after World War II, there were a lot of GIs that went to school in the GI Bill.
Exactly.
And so the universities grew quite a bit. A lot of those guys worked in for the Forest Service, like for a summer job, doing white pine blister rust, rabies
Sure.
eradication
Gil,
and stuff
Gil
like that.
did that.
Yeah, a lot of
Bob
them did.
Gilbertson.
And those guys populated all the forest pathology positions when I was going to school. And they were just starting to graduate, or to retire then. So there were going to be all these jobs. I mean, it was the height of forest pathology. And then as it turned out, as those people retired, they weren't getting replaced. You know, there was Reaganomics, the U.S. Forest Service was cutting back. They weren't hiring forest pathologists. The universities, if they had money for positions, they were hiring people in what was called biotechnology then, you know. So compared to chainsaws, this seemed like
So
a new
much for the
future
forest.
direction, you know. So all the forest pathology jobs dried up. So by '82, it really looked like there would not be any jobs. And so I was looking to change careers. Like, so maybe I'll go to do a postdoc in agricultural plant pathology instead. And then a job opened up at University of New Hampshire it was a mycology job. So I had had some pretty good mycological background at Washington State, and I got to teach a class at Berkeley when Fields Cobb got really sick. He got bit by a brown recluse spider
gosh.
and was hospitalized for quite a while, which is pretty bad luck for him, but it was pretty good luck for me because I had to carry on all this teaching stuff. Otherwise, I wouldn't have I'd had some TA experience, but here I got to teach a course. And so University of New Hampshire hired me, and that job I could kind of turn into whatever I want. So I had to teach a lot of classes, but for research we could do anything. And I did some work on a red spruce decline. It was thought to be caused by acid rain, but a lot of it was an abiotic disease. So we did that work, and then we did also start work with armillaria and heterobasidion genetics. So we progressed from just phenotypes to now we could do like isozyme analysis in the early 80s and stuff. So we used those markers for a number of genetic things. So I just kind of kept gravitating more towards kind of fungal genetics. And then things were going pretty well there, and then PCR was invented, right? Mid-'80s.
Right.
And I thought, this is going to change everything. And it did.
Yeah, it did.
So I could see the writing on the wall. I think we had you up, Meredith,
right
would
about
give a
that
seminar.
time, yeah, when I just started
And you sprained your ankle
skiing.
cross-country skiing. But outside of that, it was a pretty good trip.
I was wondering if that's where I left my ski boots. I never found them again after that trip. But anyway,
or you probably threw them away after. So we had a good time, and I think you'd been out to Berkeley already with John Taylor's and Tom Bruns were out there.
PCR.
Well, a lot of people did, right? So if you were going to learn how to apply PCR to fungi, Berkeley was the place to go. And so I got a sabbatical to go out there in 1990. So it wasn't right at the forefront of PCR, but it was in the first 4 or 5 years. Yeah, well, more for fungi. So I went on sabbatical, and then in the middle of the sabbatical, I took a job as department chair at Iowa State in the plant pathology department. So that was kind of a big jump, but I thought if we're going to do the PCR and get into more advanced fungal genetics, I needed to be in a bigger place. And still, there were no forest pathology jobs. So really, if I was going to go to a bigger place, I had to do some sort of administrative stuff. So that's what I did. So that was a pretty tumultuous time. So we had a household in New Hampshire, another one in Berkeley, and we moved from both ends of the country. Caitlin, our firstborn, was a year and a half.
Yeah, and I think I visited you when she was just a few weeks old.
That's right, really young. And then Megan was born out at Berkeley, so she was only 1 month old when we moved. So we had an 18-month and a 1 month and 2 households, and then chairing a department that was kind of a mess, and it needed a lot of kind of rough reorganizing and priorities set. So that was kind of tough too. And then Bob Blanchette got me into a project where they were using an Ophiostoma pileifera, an albino mutant, to colonize wood chips. And part of it was biological control, so the blue stain fungi wouldn't hit these chip piles. So it was essentially blue stain fungus that was lost melanin synthesis. So you could biologically exclude competing blue stain fungi by applying this fungus.
And you were reducing the inoculum?
And also, and they were reducing the pitch
Okay.
in the wood. So if they went to pulp this material, it would be cleaner, so they wouldn't have to bleach it out at the end because less blue stain, and then it'd have less pitch. So all the chemicals in the pulping process would penetrate better and stuff. So it was an interesting project, and so Bob got me involved when I was in New Hampshire because they wanted an expert on Ophiostoma because they'd have to apply for getting permits and all this stuff. So after I just started in Iowa State, and they were trying to get this product cleared in Australia, and somebody objected to having an Ophiostoma because it could be a pathogen on pine trees and stuff. So they sent Bob and I down to Australia to kind of soothe the way with the quarantine people and stuff. And Glenn Kyle was the top forest pathologist down there at the time. So he was in charge of, he'd just taken a job as charge of all forestry research for the Australian government at the time. And
he was in Tasmania at first?
He was at Tasmania, and he was just getting ready to move to Canberra for as being the national
And that was
director.
about the time of the second Mycological Congress at Tampa, I think.
Maybe.
Right
I don't remember.
there.
So this would have been 91.
Oh, it was much later.
Much later.
Yeah.
Much later.
Yeah.
So we convinced the company that you should really send us to Tasmania because we got to talk to Glenn Kyle about this fungus, So
They sent you?
yeah, they flew us over to Tasmania, and we're out in the Nothofagus temperate rainforest. And Glenn and I had worked with the fungus that was killing the big Nothofagus trees. And it's actually in the Ceratocystis family.
Right.
Yeah. So we were looking at that disease, and the fungus was killing trees almost the diameter of this room, just huge trees with big infection centers.
Was that in Western Australia or
in Tasmania?
Oh, it was in Tasmania.
There's a little bit in Victoria, but most of it's right in Tasmania. So we're out there, and Bob says, you know, there's a couple of fungi that can decay nothofagus, and because of the unique chemistry, they can almost completely delignify that wood. And he goes, here's some right here. And he goes, I don't know if this is Ganoderma or Armillaria, but both of those fungi can turn nothofagus to this clear jelly-like material. And he says it's edible. You know, they can feed it to cattle, it's, you know. And so I tasted it. And it didn't have much taste, and it had a few slivers in it and stuff, but I ate it. It's interesting. And then Bob explains, he goes, nothofagus in Chile, when it's decayed by these organisms, it's consumed by this mythical figure called Cracow. And he's a little impish guy with cloven feet, you know, so he has to, with just 2 legs, he has to have a cane to walk. And he's kind of ugly looking, and he's really a mean guy. And he seduces women and always gets them pregnant. So he's like this fertility, super fertility figure, mean fertility figure, but he eats nothing but this armillaria jelly, palopatrio. And I says, you know, you should have told me before. Oh, don't worry, it's just a myth. This is nothing. So I get back, and within 2 days of my return, Sean, our 3rd child, is conceived. So it's super powerful stuff. So when Sean's born, now we have 3 children under 3 years of age. You know, and this new department chair thing, and I'm trying to keep my research, and I kept teaching through the years.
A lot of balance.
Yeah. So Arthur Kellman was president at APS at one time. He was in town. He says, well, Tom, just make sure you don't keep doing this job because you got nothing else to do, which is pretty good advice, and I knew that already. So I served my 5-year sentence and got out. So.
Did they give you any compensation for it? Did they give you a technician? Or
oh yeah, I got
students?
some, yeah. So that was part of the incentive too. So I had a technician that was covered by college funds, a little bit of operating money, not too much. And I had some grant money here and there, different projects, but for, that 5 years, it was adequate. Yeah.
Well, I like your website because you've got it grouped by organism.
Yeah, over the years.
Because you've worked in so many different, slightly different organisms.
Yeah. So you're supposed to, I was told as a plant pathologist, you should pick one group of plant pathogens and stick with that for your whole career and be an authority on it.
Yeah, or one host plant, one, let's do the strawberries.
Sure. Yeah, that's right. So you'd work on a commodity that had a growers group that had a lot of money to do research.
No, that's
and stuff.
one of the tactics.
Yeah, but that doesn't work in forestry.
No.
The money is really, really tight. And so what I found out as my career went on, there are a lot more job opportunities and research money in mycology than there is forest pathology. I never thought that when I started, but that's the way it ended up. And so the only forest pathology job I got was that summer job at Fort Collins after I graduated.
So much for your ambition there.
Yeah.
Out in the forest and doing this work.
So do you think the Canadians do more forest pathology than Americans? I've always
they don't
had
do
that
as much
idea.
as, per capita, certainly, because timber industry is so important. And I think that's still true. I think they've lost positions though also. Oh yeah, I think so. So it just isn't that sexy for funding agencies and certainly departments. So if there's a forest pathologist that retires, they're not very likely to replace them. There's a few. So over the last 5 6 years. There's been a few in the country, but not very many. It's still pretty tight.
But with what you're doing, you know, now that you're the expert of all things, you get to go. They come and get you and send you. Hawaii.
Yeah. So
weren't you working in South America?
Yeah, quite a bit. So I had a couple of NSF grants. So I guess this would be kind of late 90s. I did go down to, in the interim, I went down to South Africa and did a sabbatical with Mike and Brenda Wingfield. And Mike certainly has been able to do forest pathology. He's got a lot of industry and government support down there, so he didn't have any trouble. But here in the States, it was pretty tight. But then when I had the NSF grants to work on ceratocystis, that changed things quite a bit. So there was kind of a steady stream of money where you could actually have students and support them. And there was some department support available. Not anymore, but at that time, my department had some support too. So you didn't have to have a huge grant to be able to piece together something for students. But, so that was a lot of international work, and the most important ceratocystis species were in Latin America. So that's where we worked a lot, especially in Brazil. So Ocelino alpinus is a forest pathologist down in Universidade de Viçosa, the federal university there, and we sent students down there, and they came up to the U.S. quite a bit over a period, maybe about 10 years. So that was kind of the height of the ceratocystis work. I worked on a few other things, but then now that I'm kind of looking at retirement, I probably would have retired by now, but there was a new disease that showed up in Hawaii. It was bad for them. It was good for me. It was kind of tailor-made for me, and it's on their most important forest tree, ohia, and it's culturally very important for them too, so there's a lot of interest in it, and so I got in as the ceratocystis expert.
I'm assuming it was introduced from...
Yeah, it was introduced. Phylogenetically, it's closely related to Central American strains or Caribbean strains. We're not sure where. It's pretty closely related to a coffee pathogen in Costa Rica. But there's some others around the Caribbean that are also closely related. So there's a Latin American clade of ceratocystis. That's where the major aggressive pathogens are. And then there's kind of a Caribbean subclade and a South American subclade. Actually, they overlap in Northern South America, but this is in that Caribbean group, which we hadn't studied as much as the South American strains. So probably on some nursery plant material.
Yeah, things got moved around.
Well, so it showed up around Hilo, which is on the east side of the Big Island, and that's where their nursery industry is. So most of the stuff distributed throughout the islands is propagated in Hilos, and Hilo's taken in stuff from Asia and a lot from Florida. A lot of material comes in from Florida, and so it could have come in on something like that. We're not real sure, but we know it was an introduction. So just to show you how far fungal genetics has gone from when I said we were just working through phenotypes, right? And then we had isozymes, we were doing RFLPs, and then PCR came along so you could do some sequencing and AFLPs and different markers. And now, you know, it's pretty cheap to sequence the whole genome. And so with this Hawaii one, we've gotten money where we can do these long read sequencings, like PacBio sequencing, and we can get the sequence of whole chromosomes, right, from one telomere to the next. You know, I mean, it's just incredible. I would have never guessed that was possible even 15, 20 years ago, but now it's routine. So we're doing that kind of analysis with the fungus. I think we're getting a handle on a gene cluster that's real important for ceratocystis, for the unique type of diseases they cause. And then we doing genetic markers to see accumulations of mutations. So it may have all started from one spore. That's what the analysis suggests, and how it spread out over the island. And then It's got a unique way of dispersal. This Latin American group, the Ceratocystis, is it goes systemic through the woody host and forms dormant spores, Allurioconidia, in the wood that can survive for years. And they stay in there unless a chainsaw cuts through and releases all those spores to the air, or ambrosia beetles will hit these dead trees after they're dead. And the ambrosia beetles don't eat wood, they just chew it up and push it out externally to have a clean tunnel for their symbionts to grow in and sporulate. So it's in that ambrosia beetle frass. And a lot of people wouldn't believe me, but I said, this is what we've seen throughout South America with this one Latin American group. The spores are really hot in that ambrosia beetle frass, and there's a lot of that frass out. So they're wood-dispersed fungi. And normally they get into the soil and then infect through roots. But in this case, we don't see the root infections in Ohio and Hawaii. It's after storms. So if they have a tropical storm go through and it appears to open up and wound these trees, they have upright branching with included bark, and so it's real easy for them to kind of crack open and the fungus gets in through there. So we've been trying out management to try to reduce the frass production that's airborne in these stands. So we go to a critical stand and then they'll cut down all the trees, get them on the ground. If there's just a few trees, they tarp them up so the frass doesn't come out. So we're trying to contain it. But it's kind of fun to apply, 25 years of experience in Latin America and then find out, oh, somebody's actually paying attention to me,
Well, and the biology of the organism, too.
Yeah.
Knowing and understanding how this all works.
Yeah.
And then you've been down in my neck of the woods, my new neck of the woods, around Savannah. And
so that was another interesting project, too. So this was with Steve Frederick, is with the U.S. Forest Service, the research station there in Athens, Georgia. And he called me up and he says, there's a funny disease showing up at Hilton Head Island in South Carolina. And I got this fungus out that maybe you might know about, because he knew I worked with bark beetle fungi and stuff. And so he sent the cultures to me, and from the DNA sequencing, we could tell it was in that Ophiostoma thales group with leptographium and others, pretty close to leptographium, but really closest to some known ambrosia beetle symbionts that hadn't really been studied too much. You had had a student, or a couple of them maybe, that had done some early sequencing and showed that some of these ambrosia beetle symbionts were in Ophiostoma.
Yeah, Steve Gassar.
Yeah, Steve, that's right. And then, so this was kind of in that group, and none of those fungi had been thought to be plant pathogens before, but this one was, and it caused a vascular wilt disease, and it was associated with an ambrosia beetle that had been recently recognized there that was Asian. And so we worked on that. We worked out the disease cycle on that. It was pretty unique, and named the disease laurel wilt, because it hit other members of the laurel family besides redbait. That was the host that initially Steve had seen. But it goes to sassafras, avocado, and all these American species in that family are just super susceptible to the fungus, and it causes a systemic wilt type disease. But it was brought over by this Xyloborus glabratus, this Asian beetle.
Yeah.
I recently read something, it was on a website, I think maybe North Carolina did it, but it was about the Chinaberry trees are resistant, and those are Asian trees that have been brought over.
Yeah, we had a study we did in Taiwan. I'm not sure if it was Chinaberry, but there were some native Asian Taiwanese.
And they were resistant.
They were resistant compared to the U.S. species. So probably the Lauraceae that are aromatic, and the ambrosia beetle needs an aromatic host, and it actually attacks a live host. It bores in, but it can't lay brood in a live tree, so it bores in. The pathogen comes out of the mycangia and inoculates the tree. The beetles back out, and find another tree to breed in, but they've inoculated that tree, and then another generation of ambrosia
looking-
beetles
Yeah,
can breed in
they
They're
that
looking
tree.
toward the future.
Create
Yeah, they're
the
knocking 'em.
neighborhood.
Yeah, Dutch elm disease works like that
Yeah.
too, where the beetles feed in the twig crotches and stuff. So,
Yeah.
so yeah, I said that it's got to have some way to bring this into live trees. And we actually looked in twigs on trees before they started wilting. We just take other
See what was
branches,
there.
and yeah, there'd be the feeding sites or boring sites, and then
Yeah.
the wilt showing up there.
So
So-
what would,
Go ahead.
I'm
No, go
just
ahead.
trying
You
to
go ahead.
bring your varied career
Yeah.
to, what do you tell your students about how they should approach their work, their life?
Yeah, be open to new areas and stuff. Be pretty broad-minded.
If you can't do forest pathology, then there's...
Well, there are people that do forest pathology and get by, and that's great to try to pursue that career. But don't pass up other opportunities. I mean, my career has been a series of accidents and things
Yeah.
that you'd expect to be disappointments and stuff, but they turned out to be really
I think
good and
that's,
fortuitous.
I think that's often the way that things happen.
Sure.
The planned life is often not the
Yeah.
one that happens. where
So
you
it was. Some people could say it's luck. It's certainly accidents,
yeah.
but I just tell them, get in the way. So when these accidents or luck comes by, you're there to take
Yeah.
advantage. So get out, go to meetings, meet with people. Don't pass up many opportunities. You get a chance to travel somewhere, do it,
Yeah.
Yeah.
and take advantage and keep your mind open, because there's probably lots of things that could excite you just as much as forest pathology or whatever you think you want to do. So I ended up doing more mycology than forest pathology. I worked on fungi attacking strictly agricultural crops too, and so forest pathology ended up being less than half.
That,
of
you
my
know, I
work,
think
you
of
know?
you as a forest pathologist
I-
because you
yeah.
study tree diseases.
Yeah, and that's what I
Even if
always
they're fungi.
wanted. That's what I always wanted to do, but I had to do a lot of other things too. And so keep open, stay enthusiastic about a lot of things, and you never know
Yeah.
what's going to happen. So I stumbled into forest pathology because the curriculum for forest biology changed, and then I just kind of drifted into mycology, and then the fungal genetics got more and more interesting as the tools went on, and so stay open. And get the background in like teaching and other things too. Don't get too fixed on what you want to do. That's what I tell students, right?
So if these tree diseases keep popping up, does this mean you'll never retire?
No, I'm going to retire or expire, as I was telling you.
I hear you're a great golfer.
No, I'm a terrible golfer, but I kind of like to do it. But I'm not good at it.
And what about politics? You
Well,
get
I used to
interested
be,
in
yeah, I
politics?
got interested pretty heavily. So George Bush got me interested in politics. After he started a second war, I decided, this isn't right. So I was pretty upset by the whole thing.
I remember you had a sign in your front yard about numbers of deaths.
Yeah, we kept track of the number of U.S. Military that died or were wounded in Iraq. And so there was a congressional candidate that came up that I supported, and then I got interested in the Iowa Democratic Party politics. So I was on the state central committee for the Democratic Party for a while. I chaired our congressional district, and I was a big supporter of Barack Obama's. And 2007 was just a huge year in Iowa then, so we get a lot of attention, and Barack Obama's strategy
and
was
you-
to win the caucuses there, or at least finish up there where he'd survive and move on, and we kicked ass.
And I think I saw your picture in that huge inaugural mass with so many people there. Did you not go?
No, I went to
Oh.
the inauguration.
Okay.
I don't
Yeah,
think
I
you saw
saw-
my picture. But
My daughter was there.
yeah, that was a great party too. So I was for about 5 or 6 years, I was pretty active in politics, and not so much anymore. I mean, I follow it, but I'm not on the state central committee anymore and stuff. Yeah.
Good.
Yeah.
So, good, well balanced.
Thank you. Yeah, are we?
Yeah.
Thank you.
Unless there's something else you want to say.
No.
No.
Okay.
Yeah.